Welcome to MOOC NPTEL course on Interactomics. Today we will talk about protein-protein interaction study, immobilization of the ligand on sensor chip. Surface plasmon resonance has transformed the study of biomolecular interactions by delivering a platform that does not require the ligand or analyte to be labeled. SPR measures the interaction between a ligand which is immobilized on the sensor chip surface and an analyte which is passed in the solution. This measurement takes place in real time, label free environment, providing kinetic, equilibrium and concentration data. In today's SPR immobilization experiment, the ligand anti-beta 2 microglobulin will be covalently immobilized to the surface of sensor chip using amine coupling chemistry. The direct immobilization of the ligand is known as direct coupling. Another way of immobilizing a biomolecule is by using capturing chemistry. In this case, the ligand is not covalently immobilized to the sensor chip surface, but is captured through the electrostatic interactions. The major steps involved in the immobilization of anti-beta 2 microglobulin antibody will involve activation, preparing for amine coupling, immobilization and deactivation. So, let us have the lab experimental session now.
Let us learn a little about the basics of immobilization and SPR assay. In the molecular interaction study, using surface plasmon resonance, we will immobilize one of the interacting molecule, that is anti-beta 2 microglobulin, on the gold sensor chip surface, while the protein beta 2 microglobulin will be passed over that surface in solution. Here, the ligand refers to the immobilized component and the interacting partner in the sample injected over the surface is referred to as the analyte. The three major steps in a Biocor SPR assay involve immobilization, the process by which ligand is attached to the sensor chip surface; interaction analysis, where the analyte is injected over the sensor chip surface and the interaction between the analyte and the immobilized ligand is monitored; and regeneration, the process of removing bound analyte from the ligand on the surface.
So this is a surface plasmon resonance and it's a Biocor T200. So this machine has the various parts, like the running buffer is connected here, and any of the biological buffers can be connected as a running buffer. They could have any pH from low to high. So the regular buffers used in a Biocor experiment are HBS, EP, HBS N, PBS for small molecule experiments. Coming here, we have a water reservoir. The water reservoir is useful for cleaning needles and syringe, and we have a waste reservoir here, and this waste reservoir collects all the waste. The samples are actually sent in this compartment. Chip is docked at this compartment. So the experiment starts by picking up samples from this compartment and transferring them here at the interaction site, and then the experiment is recorded on the screen. Here are some kind of indications are provided. When the machine is ready or the system is calibrating the temperature, when the new chip is docked and the run is actually happening. So we will now connect a new running buffer here and prime the system before our immobilization experiment. So here the new HBS EP plus buffer is connected and the new chip will be docked now. So let us look at the chip now. So this is a new chip. The chips are generally provided in these cassettes, and the new CM5 chip will look like this. This chip will be inserted in the chip docking area. From the control software, we will eject the sensor chip that is connected. So now the old chip will be removed and we will insert a new Biochemical chip CM5 here, and the insertions or the way orientation of the chip is shown on the chip in arrows. And we will close the compartment door and identify the chip from the chip type here, and the chip will be given a new ID. And sometimes it's very essential to add the name for the chip and also the lot number. And say dock chip. Now the chip is getting docked. So we have connected a new buffer. We will prime the system. Priming is a process of sending buffer through the IFC and equilibrate the system before our experiment. Generally, in any Biochemical experiments, buffer should be connected and equilibrated overnight, or if there is no time, a minimum of 3 hours of equilibration is essential. Otherwise, when to start a new experiment, do at least 6 primes on the system. Now that the chip is docked, we will prime the system. Priming, we have connected the buffer, so we'll just say start. And it takes 6 minutes for the system to prime. Now the prime procedure is complete. We will do an immobilization of a ligand. In this case, today we are actually immobilizing anti-beta-2M antibody. For that immobilization, let's prepare a wizard
Before we set up an immobilization protocol, let us understand a little about immobilization levels. The binding capacity of the chip surface will depend on the levels of immobilized ligand. The term maximum response, referred to as Rmax, is described as the binding capacity of the surface in terms of the response at saturation. A theoretical Rmax value can be calculated using the formula shown below, where RL is the immobilization level and Sm is the stoichiometric ratio. A theoretical calculated Rmax is often higher than the experimentally derived Rmax for the same interaction. This could be because of several reasons, such as the ligand is not fully active or that there is steric hindrance in the interaction. Different applications may require different binding capacities and thus different immobilization levels. A low Rmax is often beneficial in kinetic analyses, while higher immobilization levels are advantageous in binding analyses and concentration measurements. Today, we are going to immobilize the antibody on a CM5 chip using amine coupling chemistry, and the figure here shows a typical immobilization sensorgram using amine coupling. The three major steps involved here are activation of the surface esters using EDC and NHS, covalent coupling of the ligand on the sensor chip using amine groups of the ligand, and deactivation of free esters with ethanol amine. We will analyze the results of anti-beta 2 microglobulin immobilization later in the lecture. Reference subtraction is particularly important for assays where measurement is taken during the sample injection. The bulk contribution due to any difference in the sample matrix and running buffer can be subtracted by using a reference surface. This reference surface is typically placed upstream of the active surface. The flow cells on the chip surface are optimized accordingly for use in pairs, that is, flow cell 1 with flow cell 2 and flow cell 3 with flow cell 4.
File, open new wizard template, identify immobilization from surface preparation, say new, so we will immobilize for binding, flow cell 1 and 2 for binding and 3 and 4 for kinetics. Prime before run, normalize detector, say next, and the rack positions are displayed with the number of vials and the volumes and the positions here.
As was listed on the table, We will now have a closer look on the buffers and reagents required for immobilization of anti-beta-2 microglobulin. The reagents include a stock concentration of anti-beta-2 microglobulin, from which a working concentration of 30 microgram per ml will be made using an immobilization buffer of 10 millimolar sodium acetate pH 5. We need HIPIS EP plus pH 7.4, which will include 10 millimolar HIPIS, 150 millimolar NaCl, 3 millimolar EDTA and 0.05% P20. This will be used as a running buffer, which is already connected to the system, followed by priming of the system. EDC and NHS in the amine coupling kit are used in 1:1 ratio for surface activation. Lastly, we also require 1 molar ethanol amine HCl pH 8.5 for blocking the free ester groups on the surface. We shall now proceed to use the above mentioned reagents for our immobilization experiment. We will now work on the reagents required for the immobilization of anti-beta-2 microglobulin on the sensor chip surface. We will first dilute the stock concentration of the antibody, which is 1 mg per ml of anti-beta-2 microglobulin, in 10 millimolar sodium acetate pH 5 to make a working antibody solution of 30 micrograms per ml. For this, we will take 6 microliter of the ligand stock and mix it with 194 microliter of sodium acetate pH 5. The choice of the correct immobilization buffer is an important parameter to consider, and the pH scouting feature of the system can help in choosing the correct pH of the immobilization buffer. This is our 30 microgram per ml of ligand solution. Aliquots of NHS, EDC and ethanolamine are prepared and transferred to the specialized tubes used for the system. We have now transferred all the reagents into these specialized tubes. So we have 2 NHS tubes, 2 EDC, 2 empty tubes for longer stability of EDC NHS, which will be mixed inside the system, 2 ethanol tubes, one for the blank flow cell and the other for the active flow channel, and 1 ligand solution. We will be using HIPIS EP plus as the running buffer, which will be connected to the system prior to the initialization of the immobilization run. We will now proceed to insert these tubes into the appropriate rack and then into the system for immobilization of anti-beta-2 microglobulin on the sensor chip surface.
Now that we made the template, we will insert the vials at the right positions. We just fill them with the required volumes of different chemicals. To start with, the EDC is positioned here, the NHS is positioned here, the ethanol amine is positioned here, and there is an empty vial. In a similar way, another row is also made, but so one for the reference surface, another for the active surface. To normalize the chip, normalization solution for the machine to normalize the discrepancies on the RU responses on 4 different flow channels. We close the door this way and go on the screen here. We eject the rack we will insert the rack into the sample compartment this way and we need to lock it inside and then go on the screen close the compartment. We go to the next tab. So here there are some points we need to take care before start of the run. Make sure the correct sensor chip is docked. Make sure all sample reagents are loaded in the rack and microplate according to the rack position setup. Vial should be sealed with caps. Place the buffers, place water and make sure there is sufficient amount of water and buffer. So once we go through all these and everything is set in the machine, it shows the estimated run time as 1 hour 3 minutes and we have connected the running buffer. Now we will start the experiment. This will prepare the chip for the immobilization process and immobilize the antibody or the ligand of our choice today, the anti-beta 2M. We need to save this as, and now the immobilization process is running.
We will now look at the results of our anti-beta 2 microglobulin immobilization performed on a CM5 chip. Looking at the immobilization results dialog box, there are 2 response levels calculated from the sensogram. The response bound, which represents the amount of ligand bound to the surface after ligand injection, whereas the response final best represents the amount of ligand covalently bound to the surface. So here, as we observe, 12,894 RU of anti-beta 2 microglobulin is immobilized on the surface of the chip. Now analyzing the immobilization sensogram, we observe the baseline here followed by EDC NHS activation of the dextran matrix, which is again followed by baseline after activation of the surface. This is followed by covalent coupling of the ligand to the dextran matrix. The buffer washes away the loosely associated ligand molecules. Deactivation and further washing away of loosely associated ligand happens, and the difference in response between these 2 points reflect the amount of anti-beta 2 microglobulin immobilized. So, as we observed, we have successfully immobilized anti-beta-2 microglobulin and will now proceed for our binding experiment in our next lecture.
Performing an interaction analysis on an active and stable ligand surface is key to generate robust data set. We have witnessed this procedure for immobilizing anti-beta-2 microglobulin antibody on a CM5 sensor chip surface. In next lecture, we will talk about protein-protein interaction study, binding analysis. Thank you.